So here's the introduction. Maybe I'll start out by tossing a question to all the panelists who are here today about this fascinating research that basically, what's the most fascinating part of your scientific research? So, anyone can get started.
I'll start.
Yeah, go ahead.
Yeah, so hello everyone, my name is Stephanie Sang. I think some of the most interesting aspects of my research is that I study multi-trophic interactions. So I study interactions between a bacteria, an insect, and a plant. And I think those very different organisms make my project very interesting.
Okay, when you finish, you can call on another person. So go ahead, Stephanie.
Jess?
Hi, everyone. My name is Jess, and I am a second-year master's student at San Francisco State, and my project aims to train the immune system to recognize cancer. So I'm studying 2 receptors, and it's really interesting because growing up, I didn't know that you can reprogram cells to behave how you want. And I think this is a very exciting part of my research, being able to reprogram T cells into a more therapeutic cell that can actually eliminate disease such as tumors. And I will popcorn it off to Jan.
Thank you, Jessica. So I am studying a virus that causes cancer called KSHV. It's a type of herpes virus, but in particular, our lab is studying the role of one receptor that the virus encodes. And what's interesting about this receptor is that it's actually very similar to a large family of receptors that humans encode called G protein coupled receptors. And what I find most fascinating about this research, aside from the fact that I never thought I would get into virology, is just the constant parallels between humans and viruses, which you wouldn't even think of right away. We often hear that pathogens try to mimic their host tissues so that they evade the immune system, but I never even considered that a virus can actually hijack or copy our homework and steal a receptor from us and have its function modified to even cause cancer. So yeah, I will popcorn it to Nick.
Great, thank you. Hello, everyone. Yep, my name is Nick, and I'm studying a population of spinal interneurons. And what I find most fascinating is the potential applications of these neurons, sort of as next steps beyond my project. The neurons that I'm working on are excitatory and have been proposed for use in therapies, so transplanting them into injured spinal cord for spinal cord injury, but they can also be used potentially in vitro to model interneuron, motor neuron interactions in ALS and other motor neuron diseases. So I'm really looking forward to future research with that. And going to popcorn to Madeline.
Thanks. Yeah. Hi, everybody. Instead of going on the hardcore science side, I'm going to go a little bit on the sort of meta level of what I think is interesting about my research, and that it's sort of the interaction between science and society that I find interesting. So I'm studying sex differences. And I've noticed as I've done literature reviews and things like that, that even what type of science gets studied is really dependent on who are the people studying it. So for a long time, sex differences weren't really studied because a lot of times men were the ones doing the research. And even when you look at the animals used, like it was mostly male mice or like male whatever organism, male humans even. And then even now, like we think about sex differences, but like how does that relate to gender, and how like gender isn't binary. So that's what I find really interesting about my research. And I try to think about the science and how it applies to society. And I'll send it over to Sarah.
Hi. What you said was really interesting, Mylan. I've read some of those types of papers, and that's really cool. I'm a third-year PhD candidate at UCSF, and what I study is how a class of fungi that can live in the environment and can also live inside us and cause disease can sense that it is inside us or sense that it is outside and modulates its behavior based on that. And what I find really interesting is the further I get into science, the more complex I realize microbes are. When you're growing up, you know about germs, but you don't really think of them as being like alive and able to do things and modulate their behavior. And I think it's really, really interesting how very, very thoroughly they can modulate their behavior in response to various environmental stimuli. And I will popcorn to Oscar.
Thank you. My name is Oscar, and I study how a milk supply is built with every pregnancy. And as we were talking a bit earlier, this is something that can be taken for granted, yet all mammals depend on, a milk supply to feed their offspring. So in terms of human health, there are no current treatment for milk insufficiency syndrome, and this sort of affect multiple women, worldwide. And it would be fascinating to get into the biology and be able to help those women that cannot produce enough milk to feed their young. And I'll offspring to my other fellow slug, Jordan.
Thanks, Oscar. Hi, everyone. My name is Jordan Kitts. Again, I'm also a graduate student at the University of California, Santa Cruz in the microbiology department. And my research works on formation of biofilms, which are these multicellular microbial communities that encase themselves in a self-produced matrix and live along surfaces. And I think what I find most fascinating about this research is this multicellularity idea behind them. When I first started studying bacteria, I would think of them sort of as a much more basic single-celled organism with a lot less intelligence, as you would say, I guess. And it turns out that not only are these microbes incredibly complex, as was mentioned earlier, they do these cellular processes that are very dependent on each other and work with each other to create these unique and complex different areas that have diversity. And as you go throughout these biofilms they form, you'll find parts at the bottom are way different than parts on the top. And in the end, instead of being a single-celled organism, they actually are existing as these microbial communities where there's these huge levels of dependence, and production of something by one organism can shift the entire dynamic of the community. And so that really fascinates me. And I'll pass it off to Abby.
Thank you. I will mention that I'm a first year, so I haven't had much time to marinate in a project that I can really call my own at this point. But the project that I've presented for ARCS this year deals with a copper transporter that may have a role in this iron-dependent form of cell death. And as I've moved throughout my 3 rotations now, what fascinates me the most, it seems to be a common thread in the various systems I've used, and that I haven't really thought about before getting here, has been just the ubiquity of metal ions in biological processes. It's something that, like, I never really thought to really look at, like, how does an enzyme do what it does? A lot of the time in biology, it's 2 metal ion catalysis, and it looks very simple from a diagram. You have all the residues coordinating this metal ion, and it's just so much more complex and simple at the same time than I ever thought it could be. But it's kind of haunted me throughout my rotations as each time I learn, wait, more magnesium involved in this process than I thought. Wait, iron has a role in this. Wait, copper as well, maybe selenium. And it's really had me taking my vitamins a lot more seriously these days. But I've lost track of who's gone so far. So if anyone else has anything to say about this question, unmute.
I think maybe everyone has gone. We actually have a few questions from the audience. Maybe we'll turn to that. So Madeline, here's a question for you. Can you use CRISPR to determine or change gender?
Oh, wow. Okay, that's an interesting question. So yeah, I think about sort of the differences and distinctions between sex and gender a lot in my work. So like sex, we often think of as like more biological, and then gender is kind of based on your society and your culture. That's how gender is determined. And it's sort of like a psychological, like it might be what you feel inside is how gender is determined. So if I take the question as like thinking about biological sex, it's still quite complicated because you might think of sex as being determined by what chromosomes you have. So if you have two X chromosomes, female, and X and Y male, but it's actually more complicated than that because people can have more than just those chromosomes. So you could have like 3 X chromosomes, or you could have 2 X's and a Y. So sex actually can get really complicated. There's other like hormonal enzymes that can change sort of the sex characteristics that you have. So yeah, for certain things, like you may be able to use CRISPR to edit some of those like hormone genes or things that might change sex characteristics. I think it would be pretty hard to like change the entire chromosome with CRISPR. Usually it's just like targeting one part of the DNA. But you may be able to use, there's ways of using CRISPR to detect like what type of genetic material you have. So you could use that to figure out if you have like X chromosome genes, and that would tell you, or Y chromosome genes would tell you if you're more likely to be male. So yeah, really interesting questions. And whether or not it's like ethical to sort of try to change someone's sex is like another question.
So we'll have a follow-up question, presumably to the last question, Madeline. So I'm going to put you on the horn for one more. How would sex define, and I don't know how to read the word, gonadotrophy? I don't even know what that means.
Yeah, I'm not sure actually.
Okay, so we'll give you a minute to Google
I'll
that.
look it up.
Yeah, look it up, and then I'll ask Stephanie the next question. So Stephanie, there's a question for you that basically says, you know, since there are multiple facets to your research project, which one do you consider your favorite?
Yeah, so what I really enjoy about my personal project is that there are different facets to it. So it involves field work, greenhouse work, some wet lab work, microscopy, and computational bioinformatic work. And I would say that probably my favorite would be like the wet lab work. So where I could sit at the bench and just go crazy with the pipette.
That's kind of like a meditation, isn't it? Sometimes. All right, Madeline, back to you.
Yeah, so okay, quick Wikipedia search. So it's used more in insects actually, when you have this like half male, half female form, like in morphology and what the animal looks like. So yeah, I study mammals, so that's why I'm not super familiar with that. But in terms of like how sex could influence that, that's interesting because like I know in fish, your environment can actually shift your morphology to male or female depending on, you know, like what fish are around you or what the temperature is and things like that. So I could imagine that there's like certain mechanisms in these insects that would allow them to shift their male or female characteristics. And it may not be like based on their genetics, but maybe based on their environment. But yeah, I'm not super familiar with that.
So we have a little bit of time left, so I will ask for the rest of the panelists to volunteer. How have you basically changed your work habits, work style, you know, during the COVID? Are you back in the lab now? What do you see as some of the challenges that you have? I saw Jessica walking around in her mask a little bit earlier. So, you know, whoever wishes, this is open time, so whoever wants to comment on that.
I guess I'll say a little something about that. So COVID has been pretty difficult for our lab. So we work on a system where our specimens only occur annually. So they only happen once a year.
Wow.
And that happens in the summertime. And because that was like pretty much peak COVID, or, you know, when schools were shut down, so labs were shut down also. And so we needed to have our specimens shipped to us where we normally would go out to the field, and our field site is in Minnesota. So we normally would go out there, collect our specimens, and bring them back to San Francisco to do work in the lab. But because of COVID and travel restrictions, we ended up connecting with the professor out in Minnesota who agreed to ship some of our specimens out to us. But because of the lab shutdown and everything, it was pretty difficult to handle. So part of me feels like I pretty much lost a field season and most of last year of wet lab work.
Oh dear. Grab onto this summer. Anyone else?
Yeah. So what's interesting with our lab is that my PI, Dr. Sanchez, has been working really hard to just set her lab up because she is a new faculty at SF State. She was actually hired when the pandemic started to roll around. So starting a new lab, let alone starting a new lab in the middle of a pandemic, was very difficult. So we've had to shift gears a little bit and postpone certain projects, but a lot of us are essentially working double time to kind of catch up with all the missed opportunities the past year.
I have a silver lining for this. There's tons of things that, tons of ways in which the pandemic has made lots of research more difficult. But in my lab, we've had to be at reduced capacity. I imagine that a lot of you guys are as well. And because of that, we have a very complicated sign-up system for who can use different equipment, different rooms at different times. Probably most of you are familiar with that as well. But it's made me need to plan a lot more carefully. And I've always planned to some extent, but I've had to plan a lot more this year. And I think that that's probably a good skill. I think I've gotten better at planning, and it's one silver lining of all this.
Oh, that's excellent. Thank you, Sarah, for offering that.
I can follow up on our silver lining. I guess as ordering reagents for the lab, some of them were backed order for months at a time sometimes. So it sort of forced to adopt a more green culture in the lab, reusing conicals or little things like that. So now it seems to be a lot more greener too. So.
Oh, that's excellent. Jordan, you had your mute off a second ago, so I'm going to call on you.
That was a good catch. Yeah, I was actually following up both of the previous two comments. Yeah, I've definitely gotten a bit better at planning things earlier on, and also the benefit of having all these online calendars for different equipment and things is that if I am at home and not in the lab at the time, I can still do a lot of planning, where previously it became a little bit dependent, since we're stuck on, say, physical signups, where I would just have to be sort of planning it as I was in the lab. And our lab is also goes through a huge amount of tips. We've been out of tips for probably a whole. We've been backordered on tips for almost this last month, considering how many we fly through. And so we've just gotten very good at finding alternative solutions as well. Like maybe you don't need to even use a pipette tip for this, or maybe there's some workarounds. So a little bit of creative solutions that you might not have thought of normally.
Well, that's really interesting. How do you pipette without pipette tips? I'm just curious.
That was maybe not the best example, but maybe on some bigger volume things, you don't need to use a serological pipette or something along those lines. Maybe stuff you can know that doesn't need to be too precise. You can sort of just pour it or do things like that.
Oh, that sounds great. Abby, Jessica, Nick, any thoughts?
I think going off Jan's sentiment about missed opportunities during the lab shutdown, yeah, I think like once 25% or 50% capacity hit after a few months, it really forced everyone in my lab to prioritize what time should be spent on which projects. So it took me a while to start collecting data for my own project, but a more high priority project was finishing up reviewer experiments for a paper that was finally accepted this year. So that was really exciting. But I guess the downfall of that was that I really had to really get down on my planning and conduct all of my thesis research in like under 3 months. Finally did that. But yeah, I think just being able to plan is not an individual thing. I think it really helped me connect with other members in my lab to see who is in the mornings, who could help just as a collective. And I think Abby had her mic on and off.
yeah, I'm trying to keep with the positive theme, silver linings or whatever, but I think the very stressful circumstances that we're all under right now, and everyone is, but it's allowed me to see certain sides of the people that I've been rotating with in different labs, and then people in my cohort, first years, and really get a glimpse of who they are, really. I don't know. It seems like there's a tendency, at least early on, for people to try and be like, yeah, we're a great lab. Everyone's happy. But now being able to go into the lab and interact with people on a day-to-day basis, you know, they're not feeling great, but they're still able to perform, and they're still able to mentor, and they're still willing, and they want to donate their own time to help train someone who needs a lot of training. And that has really been very inspiring for me, because I don't know if I could maintain the same level of happiness, but it's been great.
Awesome. Nick?
Yeah, I think when a lot of lab work stopped, I was someone who was in lab all the time. Cells needed feeding every day. Stem cells are the worst. And I was doing a lot of stuff at the bench, but I didn't have a lot of time to really like sit back and think what my project was going to be like in a year or 2 years or 3 years. And so, yeah, the silver lining for me would be getting the time to step back and really think about what I wanted to do and the most effective way to answer the outstanding questions that I had. So it definitely slowed down work a lot, but I think it made me work a little bit more directly.
Excellent. Yeah, so I just sent out a last call for questions on the chat. So thank you all very much, for your time and for showing the results of a very interesting work and putting the posters together and attending this session. So it has been just really fun for me to see all of the work that all of you are doing, and looking forward to seeing your names pop up on the next paper, and look forward to reading, Jessica, your paper that just got submitted. It's really excellent work. Keep at it, folks.